a topic that has become near and dear to our hearts. And I'll be introducing the experimental rationale for the NeuroCOVID-19 study based in Toronto. This picture here depicts the human pulmonary artery endothelial cell. Now, I'm not an expert on this, but there seems to be many opportunities for this pesky virus to grab onto cells that express the SARS, the ACE2 receptor. Now, as I advance my slide, this is the COVID-19 is the viral illness associated with SARS-CoV-2. The previous cousin, of course, is the 2008 SARS coronavirus, which is a reminder, 80% identical to the current coronavirus. The virus has invaded our lives, and this is reflected in the dashboard that I check on a regular basis at Johns Hopkins, where there's over 200 million people infected, and 8 billion people, 8 billion doses have been administered for the vaccine, which is clearly helping to stem the tide and is a winning proposition. Now, you may be wondering, what's the deal with this virus? How does it get into our bodies, and how does it Xerox like the world's fastest photocopy machine? Well, indeed, that is the case. Each person that is infected will, upon peak infection period, will accumulate roughly 1 to 100 billion copies of RNA of this virus. In the lungs, in particular, where this virus is the epicenter, we know that there will be millions of infectious units. However, the calculations that were conducted in this PNAS paper earlier this year, they actually did a back of the envelope calculation, fairly sophisticated one, and they surmised that the total biomass of this virus is about the weight of a sack of potatoes, 10 kilograms. So it's extremely sobering to note that without innate immunity for this virus, it has literally brought us to our knees. I draw your attention to this very well executed video from the Vaccine Makers Project. And as I shift to my next slide, I wanted to think about brain specifically now. What are the mechanisms and how solid are the findings to support the notion that the COVID-19 can impact the brain directly and indirectly? In this particular paper, it's quite interesting that circa 2014, so people have been thinking about viruses and the central nervous system for many, many decades. And the authors quote that a range of pathogenic microbes make their way into the central nervous system, of which case we see here locations where viruses can permeate and infiltrate possibly into the brain, the choroid plexus, the neurovascular unit, the blood-brain barrier, the olfactory bulb. So this is 2014. People have been thinking about this and reporting these nice figures. So where are we now in terms of the evidence, COVID and the brain? One of the most poignant papers was published in April 2020. Actually that's when we actually started this study, is on April Fool's Day of 2020. This particular paper, also April of last year, in the New England Journal of Medicine, they show in this table 5 participants that presented to the hospital with acute stroke. Take note of the age of these individuals that are, 30s and 40s. Also take note, highlighted in the blue text here, that or the blue dash line, that 2 of these individuals presented and they actually did not have any COVID symptoms at all. So the frailty or vulnerability of the brain's blood vessels is underscored by this important early work. Now, fast forward to February 2021, another paper emerged by Nannoni and colleagues. And this is very illuminating finding with respect to the vulnerability of the brain's blood vessels. And what they found in comparison of across 61 studies and over 100,000 patients that had contracted COVID-19, what they found is that individuals with certain risk factors, pre-morbid, are much more likely to succumb to having a stroke if they contract COVID. So hypertension increases the risk of stroke by a factor of 5 if you get COVID-19. The severe COVID illness also increases the risk of stroke by a factor of 5. So these numbers cannot be ignored, as you can imagine. And the pooled incidence of acute stroke directly caused by COVID-19 is estimated to be 1.4%. So little under 2% of people will have an acute stroke. Is this the whole story? No, indeed, we have other mechanisms of entry or posited ways in which the virus can impact the central nervous system and the brain. This is the second idea that people have talked about, which is the olfactory bulb route of entry. Across the top, you're seeing an interesting case study from January of this year, where a research participant had actually had previous MRI scans. And this 19-year-old female contracted COVID, and the investigators were able to look at the olfactory bulb, and what they observed is a decrease in the size of this sensory organ in the brain. Now, across the bottom, I provide 2 examples of the literature. These are quite simply heroic and inspiring studies. The first study by Meinhardt et al. Published in Nature Neuroscience. They harvest brain tissue and they look at autopsy among people that died of COVID-19 or complications. And what they are able to show, each of the data points that are shown in the graph is represented by a person deceased, and in their brain they were able to detect viral copies. Notice the Y scale here is logarithmic level of RNA copies in regions such as the medulla oblongata and other brain regions. Now, the next study by Kahn et al. Published just a month ago, they show in really a heroic study harvesting brain tissue from the olfactory bulb in the hours after someone dies from COVID-19, and they were able to identify the very specific cells in the olfactory bulb, and they noted some cells much more likely and other cells less likely to be in fact infected. So giving us more interest and findings to help us understand the olfactory route of entry. Now, the last example, I won't say very much. It's quite likely that leakage and a porous blood-brain barrier as a pre-existing condition prior to contracting COVID-19 is indeed a risk factor. This will be an underlying biological substrate for risk. And individuals that, in the case of have lacunar strokes or chronic inflammation, this would be a mode of direct entry into the brain for virus particles, as well as the body's own immune system, the so-called cytokine storm that can occur in the brain. So one supremely interesting thing I wanted to raise to this esteemed group of in this network is this notion that, yes, the blood-brain barrier is shrink wrap on your brain, nothing gets in. However, what if I told you that there's about 6 brain regions that make up what's called the circumventricular organs? Their job is to actually be porous and have openings, like an open window to the blood circulation, and detecting poisons and detecting things to warn the brain potentially. So this is quite an interesting piece of evolutionary biology of the central nervous system at play that should be taken into consideration. Now, long haul COVID. That's what we got really interested in, trying to understand the consequences of this pandemic. And there's very, very strong evidence to articulate the many factors and symptoms associated with long haul. In this study that includes 48,000 respondents, some of the most common symptoms are arguably implicating the central nervous system: fatigue, headache, attention disorder, and dyspnea, all things where we could study this with the lens of a brain lens. So that's what prompted us to create the NeuroCOVID team, which has received funding from the Canadian government CIHR. And geographically, you can see there's 3 locations and the list of investigators led by Simon Graham at Sunnybrook. It's been an amazing opportunity to create a team in very short order. And our goal is to do a comprehensive brain assessments to characterize the long haul symptoms. To do this, we are using brain MRI, and we are using other neurophysiology measurements such as EEG. You'll hear more about that soon. It's also worth noting that we have a comprehensive cognitive battery and under a detailed characterization of emotions and symptoms. This is conducted both on-site and virtually. The study design is the following. Notice this is a longitudinal study at assessments initial 3-month and 12 months. The groups to date have been individuals that are hospitalized due to COVID-19 or self-isolated due to COVID-19. All individuals have a appropriate documentation of their PCR test results. We also have a control group, which we believe is important and unique. All the controls have had flu-like symptoms or a cold, and yet they have tested negative for COVID-19. The details of this have been recently published in the Canadian Medical Association Journal. Open access and all protocol details are available for further scrutiny and for those of you interested. For those of you that have a keen interest in MRI, you'll notice that this is the MRI pulse sequence screen grab for a Siemens scanner, where we've been conducting research since April 2020. The gobbledygook acronyms here describe each of the pulse sequences, ranging from anatomical to functional, diffusion, and also contrast-enhanced scans. The data that I wanted to speak to just very briefly before handing it over to my colleagues is the susceptibility-weighted imaging. This is a 4-minute sequence with high in-plane resolution. It's very exquisite to detect cerebral microbleeds. There's interest in the cerebral microbleeds because many papers reporting now, such as this one in Radiology of 2020, where the authors scanned people that were mechanically ventilated and in the scanner, and they note that 7 out of 11 participants have lesions in the corpus callosum white matter. This is indeed what we are seeing in our case studies, which I will show here. This is a very egregious example of COVID-19 and a severe course of illness. A neuroradiologist said that there's over 100 cerebral microbleeds in this particular case, which is frankly a very dramatic example of COVID-19 having a direct impact on the brain. This is a 50-year-old male that was indeed ventilated and hospitalized. Another example here shows a similar striking finding of the predisposition of microbleeds along the midline in the white matter tracts of the corpus callosum. This is, again, an older individual that was ventilated for upwards of 14 days. A third example here helps to show our interest in characterizing changes over time. So a female in her 60s ventilated for 10 days and recovering, we're able to see the evidence of corpus callosum involvement and microbleeds and a certain persistence of these microbleeds 3 months later. So I hope I've demonstrated that it's important to characterize the direct effects of COVID on the brain. This is a respiratory illness. Unfortunately, the brain is indeed still susceptible to injury in some patients. Severe brain injury is tied to a more severe course of illness. We see that acute stroke is rare. The minority of individuals, of course, which we're very grateful for. And however, I am concerned about clinically silent brain lesions, and we should not be taking that lightly, in my opinion. The direct and indirect effects of the virus are really where we want to understand the long haul symptoms. And that's where I'll turn the floor over to my colleagues.